Welcome to Live from One Cancer Place. I'm Sarah Kelly. Emerging research continues to examine the link between gut microbiome and our immune system. Today, we will examine the link between gut microbiome and cancer. To help us learn is our own Dr. Chris Heery, Chief Medical Officer at Arcellx Incorporated, and he is joined today by Dr. Saman Maleki, a tumor immunologist and assistant professor in the departments of oncology, pathology and laboratory medicine, and medical biophysics. He is also a translational immuno-oncology scientist at Lawson Health Research Institute. Dr. Maleki's research focuses on sensitizing hard-to-treat cancers to immunotherapy by targeting DNA repair in cancer cells or modifying the gut microbiome in the host. Before we begin, I would like to thank our founding partner, Seagen, and our supporting partners, Colorectal Cancer Alliance, Natera, Colon Cancer Coalition, and Mirati Therapeutics. Dr. Chris, it's all yours.
Thanks, Sarah. As usual, I think the best way to get started is to ask Saman to maybe just run us through a little bit of his background, how he got involved in this field, where he thinks, big picture, where he thinks we are today and where we're going. I think just give us a big, broad overview and we'll dive in on some topics as we go.
Sure, sounds good, Chris. So my background training, I did my undergrad in microbiology and in Iran, in fact, and then did my master's in microbiology and immunology and in Iran and in Freiburg in Germany. It was a joint program. And then I came to Canada and did my Ph.D. in tumor immunology and cancer biology. And then I actually stayed where I was in London, Ontario at Western to do my postdoc because my mentor at the time had some very interesting translational projects with industry, and they were working on new immuno-oncology drugs that I felt that it was very, very exciting for me to participate and get to know how drugs are developed and how things are moved from lab to phase 1 trials and industry. And at the time, as I was involved in this type of research, because of my background in microbiology, I always had an interest in microorganisms. And what I realized when I was working on my postdoctoral training project, that there seemed to have been a link between microorganisms and the gut microbiome, for that matter, which is the collective organisms that exist in a person's gut. This is like the word microbiome is really refers to the genes, but it's also exchanged with regularly used as in the field, or you can also call it microbiota. And then what happened was I started to realize that there seems to have been, from some of the early observational studies, that people have looked at patients who are getting anti-CTLA-4, and you're talking about 2016. And these were the early times of immuno-oncology, really, for melanoma, if you remember. And there was some really interesting observational studies, and some of it came from Tom Gajewski's group at the University of Chicago, and they were seeing that there seemed to have been a link between response and the composition of gut microbiome in patients. And there were some early mouse studies that's showing that the microbiome was important in response to these newer checkpoint inhibitors that were coming, the anti-PD-1, anti-PD-L1 drugs. So, and then I put the 2 and 2 together, and I thought that gut microbiome is interesting because it's really a malleable organ that we can change and shift potentially. And it looks like there is something here to be leveraged for cancer patients. And then going back to my roots as a microbiologist, I started to dig a little into this, and I realized that our infectious disease team here at St. Joseph's Hospital, which is a different hospital where I am at. I'm at Victoria Hospital, where the cancer program is. The infectious disease team had been using this technique called fecal transplant to treat C. diff patients, and they were doing it for ages, in fact, as pretty much a standard care in Canada. And then I went and met with them and talked to them about a possibility of doing this in cancer patients. And they've never obviously done it in cancer patients. And, the patient population they were treating was completely different, but they seemed that this is feasible. It can be done. So, and then what we did was, had more meetings with people who might have been involved. So I went to our oncology team here and talked to a whole bunch of medical oncologists who at the time were treating melanoma patients with immunotherapy. And I can tell you, they were so excited that every time I mentioned this to them, they just walked away from me. So, everybody thought I was a little lunatic. So some people still think I am a little crazy, but so it took some persistence and some doing and printing the papers and going to the meetings with oncologists and trying to explain the data and what's happening in the observational studies and what's happening in mouse studies and how we can do this. And fast forward, it took me about 2 years until I had now a team of people who were interested to do this. And this is still before those 3 science papers came out in 2018 that really showed the microbiome can control anti-tumor response and can be very important for immunotherapy response. So, but at the time, then we had our team. We sat down with this truly multidisciplinary team of people, because obviously we were going to try to do something that was not done before in cancer patients at the time. And so we had our microbiologists, we had our infectious disease team, we had the immunologists, we had the oncologists, we had all the supportive teams. And we realized it's a very complicated thing to do because we are going to treat the cancer patients. And we were actually at the time, we're trying to do it at first line. And nobody had done it at first line, obviously. The other 2 studies that at the same time they were trying to take off, they were going to do it in the refractory setting. So we started communicating with Health Canada, which is the regulatory body up here in Canada, which is the equivalent of FDA, and finally got their approval to do this study and develop the protocol that they approved. And there were so many things that we had to work on to get this thing off the ground. And I think it was in July 2019, I think, that we finally put the first patient on the trial. It took a long, long time for us from the idea stage to actually being able to put the first patient on the trial. And as soon as we put the first patient on trial, I started to work on another study in renal cell cancer, and that's the study we call PERFORM. And the idea there was that if we can give patients fecal transplant before combination immunotherapy, anti-PD-1, anti-CTLA-4, as you know, this is a very toxic, very effective, but very toxic combination. Patients often develop colitis, and it can be very difficult for the patient. So the idea was that could we do this fecal transplant before immunotherapy and then during the epicycles so that the patients maybe would experience less colitis. So we got that trial off the ground, and then we put the first patient on that trial, and then this little thing hit called COVID. And the first thing we know that the virus can be found, SARS-CoV-2 could be found in the stool. And so Health Canada shut us down. So, and they said, no more treatment for you guys until you can figure out how you can screen your donors and can verify your FMT material that there is no virus in it. So then we got to work to see if we could effectively screen for the virus. And long story short, I realized first there was no lab in the world at the time who would do that in stool. Took us some time to develop a protocol, work with a CLIA certified lab to develop the test, and then we became the first group actually in North America that was able to reopen the trial and get also the C. diff clinics open, because that was also important. As you know, patients rely on this for C. diff, but they would not be able to open their trial. So we published that, and other people started to replicate that. And so we are here today. We finished our MIMIC trial, which is the first melanoma trial we did with 20 patients. We're in the process of publishing the data, and we have 14 patients on the renal trial. Then we opened the phase 2, where we do fecal transplant and immunotherapy in melanoma and lung patients in multiple centers. And I'm in the process of opening a study in pancreatic cancer, hopefully very soon.
That is an awesome overview. There's a few things I want to touch on just for the audience. So first thing, when you say C. diff, can you just tell everyone what that is? Because I think that's probably a starting point. Most people for at least half the audience will probably say, I don't know what that is. So
Yes.
can you just talk through that a little bit?
Sure. It's C. difficile. It's a bacteria, and sometimes when patients have some sort of infections and they were put on broad spectrum antibiotics, and this organism can outgrow the other bugs in the stomach, and it can cause this ungodly diarrhea in patients that actually can be lethal. And it, in many cases, is actually refractory to antibiotics. And although it's commonly treated with antibiotics, patients have experienced a lot of recurrence. And what people have been doing in the field is that they give patients a fecal transplant from a healthy donor to completely change the biome and to make it very difficult for that organism to outgrow other organism. And that's what C. diff is in a nutshell.
Yeah. Let's make sure we're really, I guess, graphic. What is a fecal transplant? How does it work?
So a fecal transplant is, I gotta say, so it's not exactly what it was maybe a few years ago. You see, I have a lot of poop emojis and poop things that people, patients give me as gifts and stuff because we obviously deal with poop a lot. So the fecal transplant traditionally used to be effectively you would get stool from somebody who's healthy. Hopefully somebody would have done some screening of these people and for infectious agents, and then you effectively use enema and do some sort of a bowel prep initially to make sure that patients can poop out their own microbiome as much as possible, and then use an enema or a colonoscopy to effectively spray the colon. So now in our case, what we have done, our group has done, as we moved on from those, and we use capsules. So as we call them, crapsules. So for a fecal transplant. Now, how it's done is that we have established criteria for donor selection. These are healthy individuals. We have a pool of donors. They undergo a very, very rigorous screening. In fact, Dr. Mark Silverman, who's my partner on this, who's the chief of infectious disease, the chief of infectious disease department, runs the fecal transplant program at our hospital, says that it's statistically it's easier to apply today to Harvard Medical School and get admitted, accepted to the program, than become a stool donor. And that's absolutely true because we start with a large pool of potential donors and screen them for any possible metabolic disease or chronic condition that they may have, or anybody in their immediate family members may have, even if some immediate family member has a particular disease that may disqualify you for being a donor, a potential donor. And then they get tested for any infectious bloodborne diseases or any infectious agent that they may found in their stool. And even after all of that, and if somebody, and now obviously we've added layers and layers for protection against COVID. And if somebody's selected and they become a potential donor, what happens is that they still, if they decide, up here in Canada, it's cold, and in winter I want to go a trip to Mexico, that effectively puts them outside the donor pool, and they have to be re-screened again, or they get a new piercing, they come off as donor, or they change their sexual partner, all of that. So, and we actually now we screen even for monkeypox. So even before it became a big news, we were started to screening for monkeypox. So because we have to be ahead of all this stuff. And then what happens is that they come and they drop a stool for us, and the stool gets quarantined and all that because of the COVID testing. And after it comes back negative, staff will effectively prepare the stool. They mix it with some glycerol and centrifuge it down. And really what happens is that it goes through multiple processes, and you're left with the microbial component of the stool that gets packaged in 3 layers of capsules. And the capsules gets frozen immediately and kept in -80. And we are allowed by Health Canada regulations to keep it for 2 months after we freeze it. And then we start with, I should say, about 100 grams of stool. And that's what we call a full transplant for the patient. And the patient comes in the night before they undergo a bowel prep. We don't use antibiotics. As you know, there is quite a bit of data now that shows antibiotics might have negative impact, especially on patients who are getting immunotherapy. So we actually use a bowel prep. They spend some time pooping out their own microbiome the night before, and they can only have very light breakfast when they come in before the transplant. And then the capsules come out of freezer, -80 freezer. They get thawed for about half an hour. And then patients have about an hour to consume about 40 capsules, which is equivalent about 100 grams of stool to be able to receive or consider for a full transplant. And then, and as
that's by mouth.
Yes, it is by mouth. It's orally. It's administered orally. And a lot of people think that it's obviously it's a lot of capsules, but so far, knock on wood, we haven't had anybody who could not actually consume it. And I think as we age, we'll become better at taking pills and capsules. So we haven't had a lot of issues with that, thankfully. And then we allow, this is also the part that's very important, we allow for 1 week of engraftment period. So, and this was to effectively get at that point of 1 week, which we established, and I think it's now going to be a standard for anybody who wants to do this. It took us weeks and weeks of discussions between the teams and Health Canada to be able to allow, because we didn't want to start immediately treating patients because we wanted to allow for engraftment, but obviously we could not also delay cancer treatments for patients. And ideally you would have waited 2 weeks, but then there was no time. So we sat at 1 week, and we actually have data from our melanoma trial showing that patients who actually have successful engraftment are the ones who actually become responders to immunotherapy. Yeah.
So I'm sure for many people listening, they're probably a bit confused. How do you decide whether a bacteria is a good bacteria for the gut or an infectious agent? And can they be both in the different contexts? How do you make that determination, and how do you have that discussion with regulators? Maybe we can hold that last question for last. That's a little bit deeper in the weeds, but can you just start with the high level? Because I'm sure there are some watching this saying, wait, I thought bacteria were always bad. Are you saying they're good? How do you decide what's good and what's bad?
Yeah, so that's actually a very good question. And as a matter of fact, there are more good bacteria than bad bacteria. So our bodies rely heavily on our microorganisms that live across different sites of our body. Mainly, we have the most of the microorganisms in our body at our GI tract. And a lot of times we do need these bacteria because what happens is that they as we eat food, they effectively digest some of these in the process of digestion and create and generate things like vitamins and things that metabolites that we cannot create or generate with our own body cells. So they actually, we highly rely on these things. Now, you actually brought up a very important point. Sometimes the same bacteria that's very useful and it's in your gut, if they're displaced, and if now they moved somewhere else, let's say if they actually get into your blood, so they can cause an infection. So a lot of time, what really matters is that you need these bacteria, but you also need them at the right place. You don't need them to get into other places where they could now become a problem for your body or cause inflammation, because you have obviously the immune system is obviously trying to react to these. But a very important point, obviously, also about when we talk about gut microbiome is that our immune system has evolved with these microorganisms through thousands and thousands of years, and it is pretty much in sync with the organisms that live within our body. If it wasn't like that, because of just the sheer number of the organisms we have, you would think that we just die all from inflammation. But that's not the case. However, if they actually get past that gut barrier and get into the blood or other organs, it is possible that they can cause an infection, and the immune system cause inflammation, and all that becomes a problem.
Yeah, I think that's super helpful. Let me just give this little way of trying to phrase this for anyone who might not be totally familiar with these concepts. So I think a good rule of thumb is that biology is context dependent, right? A good bacteria in the gut can be a bad bacteria in your bladder. It really depends where it is. And it's sort of like, I was trying to think of an analogy, like let's take something we can all understand, like a cow. A cow is fantastic if you have a giant field and you need something to eat all the grass on the field, right? But it's really bad if you put it in your kitchen. It just doesn't make any sense, right? And it's simpler to think about bacteria as good and bad, but they're not good and bad. They just are. They do what they do, right? So there are, for instance, E. coli is a well-known bacteria. We've all heard of it. And when people talk about, oh my gosh, this fast food chain has an outbreak of E. coli, right? Well, that's not the whole story, right? It's probably a strain of E. coli that has a certain behavior that results in bacteria, because we live with E. coli all the time. I don't even know if human beings could live without E. coli. I don't think we could. So I think that's just an important thing for people to put in as you frame this stuff up. What you have to understand is that the complexity of what we're talking about today is actually trying to understand what is the right population, so in that right place to achieve this effect, right? So, we want all of the right things in the right place, but we also have to understand what are those right things that correspond to the type of outcome that we want in the clinic. So this brings me back to something you said earlier, which is observational studies and mouse studies. Can we walk through what is the difference between an observational study and what you're talking about that you took 3+ years to be able to get open, an interventional study and a prospective study, to try to understand those different terms? Because I think those are really key terms for people to understand.
Yeah, thanks, Chris, actually, for asking that. This is interesting because I was at a meeting last week in Toronto with really top oncology researchers in Ontario, and I realized even among some of my basic scientist colleagues, they don't understand the difference between an observational and interventional study. And I think that's really important and helpful for people to understand and appreciate. So an observational study is a study where a patient is receiving standard care treatment. So you have, let's say, your melanoma patient comes in, stage 4 melanoma, and they're giving the patient combination immunotherapy of ipi/nivo, which is considered the standard care. And now it might have changed with this new drug that came in, but however, let's say if they're receiving that, and as a scientist, what all you're doing, or as a clinician scientist, is that you're getting samples from the patient. Let's say you're getting blood or you're getting stool, and just looking at the changes that the body goes through, or the immune system goes through as the patient is receiving the standard care. So you're not really changing the treatment for the patient. You're just watching the patient as patient goes through this treatment, which is the standard treatment, as I said, and you're just looking at changes in their bodies, the immune system, their microbiome, their blood pressure, anything really. It can be their mood, it can be their emotion, all of that. So that's really called an observational study, which really means you're observing the patient as they go through the treatment. So an interventional study, or a prospective study that Chris was talking about, is what we have done in these trials. And that's when you're really changing the treatment for the patient. You're effectively bringing a new therapy to the table for the patient and testing the effect of this new treatment in your patient. And that's often done in, starts with phase 1 trials, where you really need to establish the safety of your treatment. And then it goes to the phase 2 trials, when you have a good safety signal. You expand your population a little bit to also confirm your safety signal and say this new combination treatment, this new drug that we are testing, this new technique we are using is still safe, but you are also maybe looking at a little bit of efficacy, see if it's actually doing something for the patient better than what you expect the standard care would do. And if that's successful, often this is run by drug companies. They run this to a phase 3 trial, which effectively you get a randomized trial where patients are in one arm getting the standard care treatment, and you're giving them this new treatment, and it's often multi-center and multi-countries and multiple jurisdictions to see if this new treatment works. So then the hardest part of these actually is the phase 1 trials, to get something from really a mechanistic observational study and something that was hypothesis, and take it to the patients, to build that, to to build that bridge and bridge the gap that exists between the basic science and the clinic. And that's often done with phase 1 trials. And that's what 3 of these 4 trials that I've built so far are phase 1s because they're new. And the primary endpoint, that means the primary goal of the study is really to establish safety. That what you have done is safe. It's not going to be harmful to the patient. And then we have obviously other objectives, secondary objectives, and those are looking at the changes in the microbiome, changes in the immune system, maybe looking at how patients are doing. And that's the difference between these 2.
Yeah, thanks for going through that. I think one thing that I like to point out to people is that when you run an observational study, what you choose to observe impacts what you learn. So I'm going to say that one more time. What you choose to collect in terms of information actually impacts whether you get the right answer to the question or not. Because you might be asking the wrong question, you might be collecting the wrong information, right? And so part of the challenge with any sort of new research, new idea, is that sometimes you don't know what you don't know. You don't know to ask the question, and sometimes it takes a few of these to get it right. So there are long-term studies that we sometimes refer to as natural history studies of diseases. And within those studies, we can collect things like stool and then analyze whether there are certain bacteria in that stool and then look at the outcomes in patients and see if there's a difference between them. So if you're ever at a major, an academic institution and someone says, hey, you're here to do your treatment, but we're asking people if they'd be willing to collect stool, or asking people if they'd be willing to get their face swabbed or their nose swabbed or come back for an extra blood draw every 3 months. This is the kind of thing that contributes to. So please consider it if you're asked to do something like that. It ends up impacting a lot of lives, but it usually takes 10, 15, 20 years. And the thing is, you might say, well, I'll never know if it helped anybody. Why do I care? It's like, well, that's how almost every major finding starts. It's some observation, someone has an idea, and then follows it and follows it and follows it until they can create an intervention. And I wanted to come back to the point you made about how long it took from first sort of making the observation and the idea to being able to enroll a patient. Because I wanted to come back to sort of the regulatory question. This is in Canada, but this type of discussion is very, very similar at FDA or with EMA or any of the local regulatory authorities in Europe or almost anywhere in the country. It'll be mainly the same process. It's helpful for patients to understand this. So what I'm assuming one of the questions was along the lines of the question we just asked around what's a good bacteria, what's a bad bacteria. Should you allow viruses to be in there? Should you not? Can you talk a little bit about how you had to have those conversations? I know probably not in the details of every single one, but how do you weigh what you think is good and what you think you want to exclude?
Yeah, that's a very important question for anybody who actually does these things, and it can become very tedious and very painful to go through these things, but it's obviously necessary because we want to make sure that everybody wants to make sure that we protect the patients. And so for us, in our initial conversations with Health Canada, was that like they were asking, how do we screen the donors? And as I mentioned earlier, we were screening donors for pretty much, this is a long list of infectious agents and pathogens to make sure that they don't have that, especially taking the fecal transplant to cancer patients, which they have they might be in a more vulnerable state with their immune system, although it's not necessarily the case for all cancer patients, where people think naturally when you think cancer patients, you think they're immunosuppressed, but it's not actually true. It's not that all cancer patients are immunosuppressed, especially if they're not getting chemotherapy or immunotherapy. They might be undergoing some local immune suppression by their tumor, but it's not necessarily that they are completely immunosuppressed. So we had to make sure that we don't, our patients, our donors don't have what is really considered an infectious agent. At the time, Health Canada was not interested in knowing that. So there were studies that people looked at more immunogenic bacteria. That, for example, there's this type of bacteria called Akkermansia, and there's data that shows that Akkermansia is helpful for, let's say, lung cancer patients if they're responding to immunotherapy. In fact, we actually don't see that in melanoma. But they're considered the immunogenic bacteria, and they're asking, so if a donor have this immunogenic bacteria. But the more important question from the safety point was that if the donors don't have, or they should not have any known pathogens. At the time where we were studying the trials, there was a case of 2 patients in the US who passed away after fecal transplant. And I think these were, they had hematological cancers, and they had I think it was the ESMBL infection. After the transplant, these patients acquired it, and we had to make sure that obviously we are screening for those agents, and we don't have it. I believe one of the studies in the US that they published in Science recently, what they did allow, and FDA allowed this, was that and they use patient donors, we use healthy donors, but what they said was that if you screen your patient and your patient has a particular infectious agent already in their body, then you can allow your donor to have the same infectious agent. Health Canada is a bit more, I would say, difficult in that regard. So we cannot have any of that stuff. So because we are obviously using healthy donors. So we screen for more of that. But that's where we are really, the conversations came down to, like, prove to me as a regulatory body that you have exhausted all these methods that are available to you to show that this person you are bringing to the clinic to get their stool from, to use it for a transplant in a cancer patient, they do not have any of these known infectious agents. Now, the one thing I got to mention is that we, as translational clinical researchers, we also deal with unknowns. So nobody knew a few months before COVID that patients could have, people could have COVID, the SARS-CoV-2 virus in their stool. So we have to obviously be always vigilant about these things. As I mentioned, long time before people become concerned about monkeypox, we started screening for monkeypox. And when the day came that FDA and Health Canada started to ask about monkeypox in the stool, because the virus can be found in the stool, we already had everything in place to send it to them saying that, look, we're already screening for it, and none of our donors have it. So that's we have to be very vigilant, and we have to be very careful of what you're looking for and what your donors would have before you actually put that material into a patient. Hopefully that answers your question.
No, it does, but I'm going to say something a bit controversial. You can either ignore it because it's too touchy of a subject, or you can let us know your thoughts. But there is a school of thought out there that there are certain infectious agents that actually drive the types of immune responses that allow us to fight off cancers. And it's not a small school of thought. These are major publications, major research on this topic. And one person who's really well known in this space is another doctor called Olivera Finn. She's at the University of Pittsburgh, has been for many years. And she is not arguing against vaccination. I want to be very clear about that. What she's saying is that some of the pediatric diseases that we had 40 years ago that we don't have anymore because of the success of vaccination, or at least they're very rare, may have triggered certain immune responses that are actually relevant in certain cancer types. And that may be part of the reason why we see some increased incidence of certain cancers that have these targets. Now, that's a very difficult thing to prove. I do not want to suggest that anyone knows these things for certain, but it is that uncertainty, the lack of being certain about what the complicated interactions of the immune response and the microorganisms that live in our body. It is that complexity that makes it very challenging from a regulatory perspective to say, I should leave this in and I should take this out, right? In fact, there have been a couple of papers in the last 2 years suggesting that there are certain called bacteriophages, or sort of virus-like agents that attack bacteria that actually impact what's going on in our gut microbiome considerably. And some of these, the presence or absence of them, are directly linked to the presence or the likelihood of certain cancers, especially gut-related cancers. So, and maybe you could comment on just that complexity a little bit. I know this is getting real deep down in the weeds, but I think when people look at this stuff, they say, well, this seems so obvious. Why don't you just give me the same thing that the other patients that responded to? And I think it's important for people to realize that every person's body also isn't going to generate the same sort of immune response to everything that's in their body, right? So we do this on a population level. We try to understand it, but actually there's a lot of complicated interactions here. Could you talk about that part a little bit?
Sure, Chris. As you mentioned, it is very difficult to prove that claim, scientifically. Having said that, we know, for example, the main trigger for an allergic reaction is the IgE response from B cells, and these are the type of antibody that the B cells in the body can produce. And when if you have an allergy, if a child has an allergy and they come in contact with the allergen, often what happens is that these cells are producing IgE. This is the type of antibody that really triggers this allergic reaction. And historically, then one might ask, why do we have that antibody? Why, evolutionary speaking, we have that antibody if it's really just causing allergies in our body? And historically, that antibody was important for our body's immune system to be able to fight off worms. So, and different types of worms. Remember, we have now clean water, at least in North America where we are, and some other parts of the world. There are still parts of the world that people don't have access to clean water. I think that's a very important thing. And humans regularly would come across these organisms, let's say these different type of worms, and that IgE antibody was really your weapon of choice, the weapon of choice for your immune system to fight this off. Now what happens is that we've cleaned up our water. We are not coming across all these pathogens, but your immune system still has it. And then what happens is that sometimes you have a bite of a peanut sandwich, and then your B cells say, "Oh my God, this is all worms." So let's go nuts around this. And so what really, I think the points that Chris was trying to make, there is this sense, at least in the immunology community, that we've evolved, our body's immune system has evolved to deal with certain infections and certain microorganisms, but we don't see them anymore. And because we have a cleaner society, we have clean water, we are able to take care of cleaning our food, we have more sanitation, and our immune system sometimes gets confused. And that's why there are at least, I think, studies that they suggest if you have a dog, it's actually very good for your health because you come across the microorganisms they have, you're exchanging microorganisms. Or if you have a better, more diverse diet, this is an important topic also in our area in cancer, because what happens is that could help with the diversity of your gut microbiome, and you can have more different types of organisms in your gut, and that can help your immune system and the response to these newer drugs like immunotherapy. And I think that's where this gets to. And from our standpoint, when I started this, my over high level thinking about it was that we know most cancer patients, if not all of them, have dysbiosis. Dysbiosis is when you have in simple terms, you have a messed up gut microbiome. That means you have lost the diversity in your gut microbiome. Microbiome, you have to think about it, is a very diverse community. A healthy microbiome is a very diverse community. Let's say like a human community. You need everybody with different skills in it. You need a police officer, you need a scientist, you need a social worker, you need all of that stuff. Now, if you, let's say one day you wake up and you say, you know what, I'm going to replace half of the professions in the society with just police officers or scientists. Do you think how well that society is going to run? Not that well. And that's exactly what happens with our microbiome. in our lifestyle, especially with our Western diet and our lack of really movement as much, and it's our sedentary lifestyle, what's one of the things that has been happening to us is that we're losing the diversity of our microbiome. And then when somebody develops a chronic disease like cancer, your immune system and the tumor actually can edit your microbiome. And that's also a very fascinating area where there was a study a few years ago where they showed in AML patients, I think, the tumor was editing the microbiome. So the microbiome, you would have this bacteria that they would produce factors that would more support the tumor growth than normal cells. Okay, so this gets worsened in a patient that's like a cancer patient. And my high level of thinking about this was, what if I go with the nuclear option of just taking your microbiome out and replacing it with somebody's healthy microbiome? And we know that the microbiome talks with the immune system, is going to hopefully jumpstart your immune system, and then it would get a good response in your patients. As a matter of fact, although we've done this prospectively in only 20 patients in melanoma with mature data, but we have about 65% response rate in those patients, which is quite fantastic. And when you compare it to single agent or one drug only immunotherapy, which you would expect between 30 to 40%. So we're getting better response rate in our patients with some really good durable responses in those patients. And we think, and we've done obviously a lot of translation studies showing that the microbiome was really, really important for them to be able to respond to this drug.
Can you talk about that last part? I think the part that I imagine for a lot of people watching this is that they're still missing is how.
Yeah.
How does that work? How does changing the gut bacteria affect the likelihood of your immune system recognizing your tumor?
And the how is $1 million question, and the one that a lot of people are trying to answer, and obviously our group is also working hard at it. So one thing that we've seen, which is very, very interesting, and I think, as I mentioned, there are 2 science papers that came out, and they did fecal transplant using a patient material in melanoma patients. But they really didn't show as much of an engraftment effect. There was claims in the papers, but the data is not very strong there. But one thing we see in our patients, I think it would be the first study to really prospectively show this, was that our patients who responded were the ones that so when we did the transplant in all patients, what happened was we got stool samples before the transplant, after the transplant, before the start of immunotherapy, and then 3 weeks after immunotherapy, which is 1 month post-transplant, and then 3 months later. And then what happened was, when we looked at the samples at the baseline level, the cancer patient's microbiome was very different than the donors and very different than everybody else. And then we did the transplant, and it looked like just right after transplant, their microbiome, pretty much everybody's microbiome became very similar to the donors, because obviously we just replaced their microbiome. And then when we looked at it 3 weeks later, which is about 1 month post FMT, what happened was some patients retained that engraftment. Their microbiome looked the same as the donors, similar to the donors, and some's microbiome changed and became more close to what it was before. And that's what we call engraftment. And then when we followed up those patients, everybody who were able to keep their microbiome were the ones that they actually responded to a drug. Okay, this is the first clinical evidence that we are talking about here in line of showing that an engraftment of microbiome can really drive response, or would be very important for response to immunotherapy. And then we did a whole bunch of obviously other studies. For example, we saw in our patients who responded, they had now certain metabolites, and these are microbial metabolites. These are things that these bacteria are producing in our gut, that they are able to very much activate the immune system, okay? And these patients who retained their microbiome, that means they kept their engraftment successfully, and their bugs started to produce these metabolites, and we could detect it in the blood of the patients. They were the ones that they were showing ongoing response than those that they didn't, okay? This was another layer of evidence. The other things we did, which was, I think it's a very cool experiment, in collaboration with Bertrand Routy in Montreal, and we have done some of this work now here in our lab as well. What we saw was that you could get stool from the patients before they get transplant, okay? and put the stool in mice. So these are, you could use regular mice or germ-free mice. That means the mice that they don't have an immune system, okay? You give the mouse the stool from the human. Now you've created what we call an avatar mouse model. These are the mice that they really have a human microbiome. They don't have any of their own. So, and then what would you do is that you give them the same, after you allow for engraftment, you give them the same, pretty much the same cancer that the patient had, like melanoma, or you can give them different type of tumor, okay? And then you treat the mice with the same drug, pretty much just about the mouse version of it, the same drug that the patient got, okay? And when we did that, we saw that the animals that they got the stool before the transplant, even if the patient was a responder, those mice did not respond to treatment. But when we got the stool from the same patient after transplant, a few weeks later, and put it in another cohort of mice, those animals become respond to treatment, showing that this change of microbiome seemed to be very important to drive that response to immunotherapy. And this is the layers and layers of translational studies that we've done to just prove that the microbiome is playing a key role in driving that response to immunotherapy.
A couple questions out of that. So one, do you map the diet of the donor for the fecal transplants? And have you tried to simulate repeating the diet in some of the patients versus not, and see if it affects engraftment? If the patient will stick to the diet that is similar to the donor?
It's a very, very good question. And so our donors, as I said, they have some, Olympic style microbiome. And these are people who are generally much, college school kids, but on very healthy diets. This is not something we told them. This is their lifestyle. They have very, healthy lifestyle. They're very athletic often. And what we haven't made any official recommendations to our patients, at least in our trials. In one of the studies, we are collecting questionnaires, questions about their diet. We do tell them when they come to get the transplant that there seems to be studies that out of Jennifer Wargo's group and Jennifer McQuaid's group at MD Anderson showing that there is a direct impact from the diet in retention of microbiome. So one of the things seems to be, sodas seems to be bad, much alcohol be bad. So we tell the patients that you want to generally keep a high fiber diet. And there was another study that was published in Cell, and they showed that, things like kombuchas or those that type of food or drink can be helpful in retaining good microbiome. We do make that kind of recommendation. It's not just something that it's a make or break for the trial. Diets, changing patients' diets, Chris, is very difficult. Often is very difficult, and people don't appreciate how difficult that is. Even in cancer patients, a lot of times somebody who has a heart attack, you tell them you should stop smoking, they often stop because they just don't want to go through that horrible experience and die. But changing somebody's diet is very, very difficult because people are just used to, this is what they know, this is how they cook. But diet seems to be a very powerful tool in changing a microbiome or keeping up with a good microbiome. There was a study, I think it was Jennifer McQuaid's group, that they did put people on two different diets. It was one that was high fiber, and there was just a regular standard American diet it was 2 or 3 weeks at a time. And then they were sampling the microbiome. It was interesting because at the time the patients were on this good diet, it seemed like the good bacteria healthy bacteria would just populate the gut. And then as they change, they would just go down. Go up, go down, go up, go down. So it seems like it definitely has an impact, but it's very difficult to keep people on it, especially as they're going through their cancer treatment and stuff.
Yeah, I wanted to make that point specifically so people could understand sort of how is it possible that there is such a difference of microbiome among people. And you can talk about this much better than I can, but I think what people may not understand is that those bacteria certain bacteria will grow more if the nutrients that they prefer are there, and the ones that don't grow as much will be because they don't have the nutrients that are ideal for them. So if you eat an all plant diet, you're going to have a different set of bacteria than if you eat an all meat diet, for instance, because different bacteria are going to be able to break down those foods differently, and therefore they will thrive. Just like, if you have a big forest and there's no wolves and there's lots of grass, like there are going to be a lot of deer. But if now you take away the food source, and they're not going to be there. And it's the same general idea. But what we're hearing today is that complexity, those different types of bacteria living, is actually dependent on having a broad and differentiated diet, or at least certain diet features, in order to enrich for those populations. Is that a reasonable way to describe that?
Yeah, absolutely. It's very important to understand that these bugs, as Chris mentioned, they're professional metabolizers. They metabolize everything, and then what happens is that they produce, they produce some metabolites that are useful for our body, but they also produce waste. And what happens is that if you're on certain type of diet, let's say you're just eating the regular American standard diet, okay, some of these metabolites that are produced regularly in your gut may actually become toxic to some of your own microbiome. They can't handle it, and that's why they die off, and you lose diversity, okay, because you're just really catering to one group of bacteria in your microbiome. But if you start to go on, a more healthier diet, if you will, a high fiber diet, and have less of things that we know can be potentially harmful to the microbiome, now you allow for those bacteria that they were in smaller numbers to thrive and become more dominant in your microbiome, and you create a better balance. In fact, I think there was a study that they were looking at some of the native tribes in Amazon, and they were sampling their microbiome. I don't remember the details of the study, but what the gist of it was that the diversity of microbiome in those tribes that they haven't come across, the modern lifestyle is extremely high. And just because they're just eating what, our ancestors were probably eating, the fruits and vegetables and sometimes meat and all that stuff, and they're not coming across, a lot of cheeseburgers and fries all the time with pops of soda. So that's really, there's this balance. But the good news, I would say, for people who want to take care of their microbiome is that, as I mentioned at the beginning of this call, microbiome is a malleable organ. Now, malleable but stable, that's something that's also people don't appreciate. We actually have done, obviously, a lot of samplings of our donors, and you wouldn't believe that how stable your microbiome can be. It's not something that you can easily completely shift, but you can, if you have persistence, good diet, you can, and exercise. Exercise can also have huge impacts on changes of the microbiome as well.
Yeah, I think that's a really interesting point. So, like, if you change your diet for a week, you're probably not going to shift your microbiome. It has to be a very consistent change over time, right? It is interesting, though, because I think there are probably people out there who have tried to switch to a healthier diet, more fiber, et cetera, and found, oh my gosh, like I'm having GI distress all the time, right? And that's partially because now your gut microbiome can't help you deal with the stuff you're eating, and it takes some time for that to shift over. I wanted to comment on a couple questions that have come in the chat. One, I want to make sure we get this correct. So the way I heard it is that you want the microbiome, so the fecal transplant, you want that new microbiome to engraft, meaning you want it to stick around. And that is associated with better outcomes against the patient's cancer in combination with treatments like checkpoint inhibitors. Is that correct?
Absolutely. You want that engraftment to happen. You want the take to happen, and you want the microbiome to stay. And that's the engraftment key. Yes.
So engraftment is good, and what that means is that the new microbiome is there, and that is what is related to better outcomes, just in case that wasn't clear to anybody. There's a question here about beta glucuronidase activity in the microbiome, and that being associated with good response and successful fecal transplants. And maybe if you can touch on that, and also then how is that related to diet and any known chemotherapeutic agents, if you can go down that path? Obviously a little deeper than where we've been.
Yeah, no, that definitely is. And I got to say that I'm not familiar with those studies, because there are now quite a bit of different studies that are looking at the presence of different metabolites or different molecules that they are higher, and you would find an association with response. And that is, as Chris mentioned earlier, that's a lot of it comes from observational studies, and that's all very important to find what is that one thing that's higher in the patient who responded. So a lot of times we need to see these, especially when it comes to observational studies across different studies, to see if it's reproducible or not. Sometimes when you see, I think the question I was looking at it in the chat box, it was about response to chemotherapy. It wasn't about immunotherapy. So one of the things I got to mention that I don't think we've touched on, but we have alluded to, that it would become important for any GI cancers really, but also any drug that's actually given to the patients orally. It's also very important, also systemically as well. Any drug that comes across the microbiome, it can be metabolized. And we actually didn't know, or we didn't appreciate the impact of microbiome on this for a long time. But now there is more understanding of that. They actually published a review article a couple of years ago and specifically touched on this subject, and saying that a lot of drugs or even chemotherapy that you're giving the patients, and if then they come across the microbiome can metabolize it, and it can have two effects really. It can neutralize the effect of the drug because it can reduce the active agent in the drug, or it can maximize it. So it is important. And I think a brilliant example of this is in pancreatic cancer, where a lot of pancreatic cancer patients have what's called a gamma proteobacteria in their tumors. So we now know there is something called a tumor microbiome, that pretty much any human tumor actually is colonized by bugs. We didn't know and appreciate this before. So this is now an accepted reality in cancer. Patient can have breast cancer, same thing. The tumor has its own microbiome. We still are not sure, especially for tumors that are outside the GI tract, how the bugs get there, but we know they are colonized. And in pancreatic cancer, as I was mentioning, these bacteria have the ability to inactivate gemcitabine. And why that is important is because that's a chemotherapy agent that is regularly given to pancreatic cancer patients. And their tumors have these bugs that actually inactivate gemcitabine. And so if you think about it, they're giving a drug to the patient to kill the tumor, and the tumor has these bugs, and the bugs eat up the gemcitabine. So the tumor is not being affected by it. And that's the rationale for us to do this fecal transplant study, to see, because there is studies that show that you could fecal transplant, and they've done it in mice, but you change the gut microbiome, but you can also change some proportions of the tumor microbiome. And we want to see if we can actually change the gut and the tumor microbiome to make it more sensitive to chemo, instead of the situation that they are right now.
This is an interesting question. I don't know if I've ever heard an answer to, but in getting colonoscopies, do you run the risk of the prep affecting the gut microbiome positively or negatively? Is there anything that one should do before or after a colonoscopy? We haven't done any studies on that, and I can't give you a clinical advice, obviously. One thing, if I were to do colonoscopy, obviously you have to undergo bowel prep, and which makes sense because you want to have a clean colon that they can see it, any polyps or any tumors there. But what people need to do is obviously go back to your good diet and try to maintain a good diet for after colonoscopy, or after. Not just that, if you're a patient and you have some sort of infection and you get antibiotics, a lot of people have experienced this. When you develop an infection and you're put on antibiotics and you feel that you're in a crappy mood, a lot of it is because your good bacteria are dead
with the use of antibiotics. And, there are studies shows that your gut microbiome can control your mood. So, and what I often do, if I have to go on antibiotics or any of my family members have to go on antibiotics, we go back on, having yogurts and, some good high fiber diets and things like that, things that it can support the microbiome really, instead of, just drinking pops and things that are not good. That's what you can do as how you maintain your healthy microbiome for any other condition. I would say that's holds true for everything, but obviously I'm not giving anybody any clinical advice here. That's what my lawyers always remind me.
Well, this has been a fascinating conversation. I want to be respectful of your time. Sarah, can I turn it over to you to close us out then?
I would like to thank everyone that joined us today, and a special thanks to our experts, Dr. Chris Heery and Dr. Saman Maleki. And again, thanks to our founding partner, Seagen, and our supporting partners, Colorectal Cancer Alliance, Natera, Colon Cancer Coalition, and Mirati Therapeutics. We look forward to seeing you next time on Live at One Cancer Place.